Welcome to Health Professional Radio. I'm your host, Neil Howard. Thank you for joining us. In this segment, we're going to be speaking with Joshua McClure. He's joining us as CEO, co-founder, and co-inventor of Maxwell Biosciences. It's a preclinical drug platform company that develops biomimetic therapeutics. Those are synthetic compounds that will mimic and improve upon biomolecules. Welcome to Health Professional Radio, Joshua. Thank you for joining us this evening.
Thanks so much, Neil. It's really great to be here.
Well, what's your professional background briefly? Give us a look into who Joshua McClure is.
Sure. I started with the Air Force Academy and from there went into classified technology stuff. Essentially what I do now as a CEO, managing budgets and contracts and working with engineers and scientists on inventing new things that help defend humanity from the bad guys. In this case, it's pathogens, especially the high threat ones, viruses, pandemics, things that we all are quite familiar with now.
Well, tell us a bit about Maxwell Biosciences. What problems are you looking to solve there?
Yeah, sure. We're essentially solving two problems. The main one is that viruses and other types of pathogens as well continue to evolve their proteins. So viruses evolve their proteins based on a competitive environment, just like humans evolve themselves competitively. And we're familiar with the various different strains of concern. Omicron, Delta, and those are household names now. And how Maxwell Biosciences solves that is we actually target the membrane as opposed to the protein. And so as a virus buds from an infected human cell, it buds through the membrane of that cell, very similar to the way a child or anybody at a party might blow a bubble, like a soap bubble. And the membrane of that virus is very, very similar to a soap bubble. It can be popped. And that's what we do, is our compounds are kind of like little missiles that pop those bubbles. And the way we do that is we mimic a key immune peptide of the human immune system. It's called human cathelicidin antimicrobial peptide, and it's literally shot out of T cells at these pathogens, and it pops those bubbles. It pops the viruses. It perforates larger pathogens like fungi and bacteria, perforates them and then essentially sticks to the inside metabolic components, ribosomes, and where the whole Krebs cycle is happening inside of a bacteria. It freezes all of that up inside of a bacteria. So a single compound then that we make is effective against bacteria, fungi, and viruses, just like your own human immune peptide is that all of your T cells use against all of these pathogens. So that's the first problem that we solve, is that what it appears like, NIH has confirmed, many independent researchers have confirmed that we have very, very close biomimicry of this key innate immune peptide that exists in all mammals. And so that effectiveness has been evolutionarily conserved for at least 100 million years. Wow. So it looks like these pathogens are not able to evolve a resistance to it. That's problem number one, is we think that we can essentially end viral pandemics with this strategy. Problem number two is that we are able to make these compounds very stable inside the body. Inside your body, your body is constantly breaking down these peptides, and they're gone within minutes. So this is why this endogenous peptide, human cathelicidin antimicrobial peptide, has never been made into a predictable or undruggable therapeutic. And so these changes that we've made through the synthetic process is that's our core technology and what makes it very stable, also shelf stable.
So what's this core technology called?
Yeah, so internally and for business purposes, we have branded the whole technology platform as CLAROMER. So the CLAROMER platform is really what the whole company is based on. In academic journals, it's called peptoids, Peptoid. because it's peptide-like. It's mimicking peptides, but it's much more similar to a small molecule. It is completely synthetic, very low molecular weight, and essentially where the side chains would be attached on a peptide at the carbon on a peptide. What we do is we essentially attach the side chains to the nitrogen on the backbone, and that makes this impervious to enzymatic breakdown inside of the body.
Now, I heard you mention viruses, bacteria, and even larger fungi as well. What about some cancers? I know that there is a problem with some cancer-fighting compounds attacking healthy cells at the same time. Is CLAROMER, is it precise in, as you say, shooting out this compound into cells' membranes specifically, or is it kind of a shotgun effect?
So it is actually it's very, very precise. So this is how your immune system works as well, right? So your immune system, remember that there are autoimmune disorders, right? Mm-hmm. But those are generally related to antibodies and to generally high levels of inflammatory cytokines. And that's what you want to avoid. This is why the innate immune system is so important as opposed to the adaptive immune system. The innate immune system is primarily based on peptide action. So there's two types of peptides that your body uses, cathelicidins and defensins. So there's many types of defensins. The human body only has one cathelicidin. That's human cathelicidin antimicrobial peptide. Other mammals, other types of species have multiple types of cathelicidins. It just happens that humans only have one. I don't know why. Maybe that's why we get cancer and elephants don't. Mm. But the key issue here is that senescent cells, so cells that have are signaling to the body that they need to go through the apoptosis process. They expose phosphatidylserine on the outer leaflet of the cell. So this includes infected cells. It includes any kind of cell that has had DNA damage or mitochondrial damage. Those cells go into an apoptotic process, and the way they signal that is they express phosphatidylserine on the outer leaflet of their membrane. That does not occur in a healthy human cell. Not a single healthy human cell does that, right? So that's how your immune system knows, and how the T cells know, when to shoot that peptide at a cell and to recycle that cell. This is also why viruses, bacteria, and fungi express the phosphatidylserine on the outer leaflet of the cell, because what it tells your body is that initially is that this particle is important but not urgent. That it's not required to inflame the body because it looks like it's a senescent cell. It looks like it's an elderly person, essentially, metaphorically, within a human society, right? You don't call out the SWAT team on an old lady, right? You help her cross the street, right? You get her to the hospital, that sort of thing. So these are essentially really bad guys, essentially spies and saboteurs that are dressing up like old ladies, right? Okay. So that's essentially metaphorically what the body is doing, and what we're mimicking is we are capable of addressing these pathogens and even completely wiping out these pathogens in animal models. We've tested this in 3 different animal types without any inflammation whatsoever. Hundreds of times the therapeutic dose, we see 0 toxicity.
So are you completely finished with testing? When can we expect this to become available?
So we are in talks with the FDA. We're moving into human trials. We just finished Syrian hamster studies sponsored by the NIH. I can't go into too much detail there, okay, but very, very promising results as far as pre-exposure prophylactic. And so now what we're doing is we're waiting on talks with the FDA to move into expedited human trials for the pre-exposure prophylactic.
Give us a website where listeners can learn more about the developments at Maxwell Biosciences.
Yeah, sure. It's www.maxwellbiosciences.com.
Just like it sounds, yeah?
Mm-hmm.
Well, Joshua, I appreciate you joining us here on Health Professional Radio this evening. Quite informative. Looking forward to our next conversation. Thank you.
Thanks so much, Neil.
You've been listening to Health Professional Radio. I'm your host, Neil Howard, in conversation with Joshua McClure, CEO, co-founder, and co-inventor of Maxwell Biosciences. Audio copies of this program are available at hpr.fm and healthprofessionalradio.com.au. You can also subscribe to the podcast on iTunes, listen in, download at SoundCloud, and be sure and subscribe to our YouTube channel at youtube.com/Health Professional Radio.